at UCSD at the Scripps Institute of Oceanography. But more excitingly, I believe in January, he'll be starting his own group as a new faculty at Occidental College. And so we've kept him in Southern California. So without further ado, okay.
Okay, perfect. Thank you so much, everybody. And thank you, Lee and Daniel, for the invitation. It's wonderful to be included among all of these early career scientists. As Ming said, I'm more of a pre-career scientist, I guess you could call it. But it's wonderful to be invited nonetheless. So I'll talk to you today about a project that, similarly to Adam, who gave a wonderful introduction to natural products, I started in my postdoc. But as a COVID postdoc, things change, you have twists and turns. And so I never really got to finish it, which is actually great news for my independent career, because now I get to take it with me and have it for my independent career. So was it on purpose? We'll never tell Brad. So I'll be moving just up the street to Occidental College. Oxy is a primarily undergraduate research institution. I won't have graduate students, but I might have a postdoc or two. So if I get money, I'll let you guys know. And I'll be looking for postdocs definitely. But part of why I wanted to share this project in particular was because it's actually a project that I did exclusively with undergrads while I was at SIO. And so this is Sam and Vivian. They're extraordinarily talented undergrads who I had a chance to work with. Got to see my COVID hair, and were really, really excellent. And I wanted to share their work and how it's going to be honestly driving a lot of the work that I'll be doing at Oxy. But before I get started with that, Oxy, before I get started with that, I thought maybe to give a quick overview of why marine natural products. My research group will focus not exclusively, but significantly on marine natural products. And I thought I'd give a quick touch on why. So while I was in the Moore lab, I had a wonderful opportunity to work on kind of the traditional application for natural products, which is medicines. So Selenosporamide is isolated from a marine bacterium. It's actually in phase 3 clinical trials for glioblastoma. It's a really, really interesting natural product, nice and small, really reactive, kind of a Moore lab classic. Things that I really didn't appreciate before I came to SIO was how you can use natural products to actually understand ecology. And so this is actually one of my favorite natural products, despite the fact that it's relatively simple. It's actually a pheromone, and it has this really neat reaction, which is spontaneous, where you undergo this rearrangement, which happens at an extraordinarily rapid time scale that's measurable to generate a pheromone in brown alga. Again, we kind of talk a lot about natural products as medicines, agrochemicals, but there are these bespoke uses that I think are really fantastic as well. And so I love this one. This is Shinnerin. It's made by lots of marine organisms. It's actually a sunscreen. And as sunscreens become continually banned over time at places like beaches where their corals are sensitive to unnatural sunscreens, I think it's really important for us to think about alternatives, and especially these types of natural alternatives. Lastly, this is kind of a new area that I think natural products is moving in, which is materials. So this is xanthomatin. Xanthomatin is actually a pigment that's found in octopi, and it's what they use to change color. It's electrochromic, so they have a voltage which changes the color of these molecules, and they're differentially halogenated, functionalized, and so you can actually get different colors, and DOD and all kinds of other people are really interested in this for coloring drones and stuff. Not the greatest, but it's a really interesting case study, I think, anyway. And so the last one is something I had a wonderful opportunity to work on at the end of my postdoc, and something I really didn't have an appreciation for being from the Midwest, but is a major problem on the coastal areas, which is harmful algal toxins. This is primesin. It's an extraordinarily potent algal toxin. It killed a ton of fish in Europe last year, and it really changed the kind of global, I think, stance on how we need to be able to do environmental monitoring. And so why part of we study natural products is to understand their biosynthesis. So this is a big, horrifying looking molecule. Thankfully, it's made by only 2 genes, at least the carbon skeleton is. And being able to tell if those genes are being expressed or not is a really interesting way to be able to target and evaluate if the potential for production of harmful algal blooms is out there in your rivers and oceans. So with that, hopefully I've kind of convinced you a little bit about the impact of marine natural products across a variety of fields. And part of why I came to Scripps was because of this professor right here, John Faulkner. John Faulkner was at SIO for a long time, had an incredibly storied career where he studied, for the most part, the biosynthesis of really interesting molecules from marine sponges. I'm sure a lot of you have heard about marine sponges as a really interesting source for metabolites, as well as the fact that recently the Moore Lab has shown that not only do their symbionts produce natural products, but the sponge actually encodes biosynthetic genes itself. And so really, really interesting. And I was inspired when I got my 2 undergrads to think about what could we do that would be kind of capitalizing on the legacy of Professor Faulkner. And so Professor Faulkner characterized over 40 native sponge species that are local to the San Diego area. And he found a ton of really beautiful and interesting, structurally diverse natural products. And I thought, maybe that's tapped. Maybe he found them all. You never know. But I thought this might be a fun experiment for my undergrads to see if they could. One of them had their diving certification, and so they could go out and collect their own samples, bring them to lab, process them, extract them, run the mass spectrometer, and then with a lot of guidance from some of the people here who helped me along the way, try to analyze their samples and see if there was something new in there. And so that's the story I'll tell today. So diving in head first, Samantha went out and collected 14 morphologically distinct sponge samples. They are really weird. This is immediately where I knew I was not cut out to be a biologist. Sponges are gross. Nobody tells you this. They're really slimy. There's gross stuff inside of them. It's really horrific, actually. But it's kind of worth it. They make a ton of really interesting natural products. And so I made them touch them. And so they did all of the touching. I did all the supervising. And we did a lot of this by activity-guided fractionation, which I think is still a wonderful way to be able to identify potentially new and interesting natural products. So they took our kind of fine dice of sponge and generated an extract with methanol. They fractionated that extract into a 96 well plate. And then we had this very simple antibiotic assay to identify any extracts that might have antibiotic activity. And from this sponge, Cleona salata, which is a native sponge to San Diego, it's a boring sponge. That's not to say that's not interesting. It bores into shells. And so it's a really interesting, actually, despite its name. We identified that this activity was due to this known compound cleonamide. I've highlighted a portion of this molecule in orange. It's an enamide. This is a relatively rare and kind of nondescript functional group. I think a lot of people just think it's 2 functional groups mashed together. It's not. It has its own reactivity and kind of interesting activity. And so I was like, this is really interesting, but known. But then when we looked closer at the data, we saw that there were lower activity samples that also corresponded to kind of lower intensity signals that didn't really immediately match to a known compound. And so I thought, well, maybe there's something new here after all, and maybe we should investigate what else is in this sponge. And so I looked in the literature. I should have looked more, but we'll come to that in a minute. And we found that Cleona are really talented producers of these enamide alkaloids. And so I've colored here all of the different enamide features in these beautiful alkaloids that are collected from mostly the Pacific, as well as Patagonia. And what's curious about enamide alkaloids is that they're broadly distributed across the marine environment. And it's not just broadly distributed in sponges. You find them in bryozoans, in ascidians, even in red alga. And I think what's really fascinating, and I think how I tricked Allegra into working with me, is that one of them has a really, really interesting ecological purpose that people think, which is Ascidia nigra, which is this tunichrome, the source of tunichromes, can concentrate vanadium in its body at something like 10 to the 7th compared to the surrounding seawater. And people think that vanadium plays a key role in kind of chelating and importing vanadium into that ascidian. And like I mentioned before, the Cleona are burrowing sponges, and so they need to be able to solubilize things like calcium, maybe be able to import it into their body to build their own skeletons. And I thought, kind of looking at the structures of the cleonamides and the selenamides, that these might be kind of metallophoric molecules. And so kind of interesting. And I thought, there's no biosynthetic origin. These are broadly distributed. Maybe this is an interesting project after all. So let's look and see what we can find. So you'll see a lot of raw data today. None of this is published. All of it is relatively new. It's wonderful that I'm comfortable enough to share this year. And if you do steal it, you're stealing from undergrads. And so you should feel bad about yourself. But anyway, so we were able to see this kind of original peak. The biggest peak in here is Cleonamide, which is the one that's known. But as we kind of, this was the expertise level that I had in metabolomics when I first started working with my undergraduates, as well as some of the wonderful people here, was I would take the little walkie man icon and kind of walk them along the spectra and kind of see what looked interesting. And so what I was able to do was, what's interesting about these molecules is they're brominated. And so I was able to kind of use my little walkie man to walk along and see which other ones have this very specific isotope pattern of a brominated natural product and try and correlate that with, are these known molecules or unknown molecules? And what's really interesting is a ton of them are unknown, a ton. As a matter of fact, the only one I could find that was known was this first one I'm going to show you. And there are ones that are giant, that are much, much bigger than the reported ones in the literature. And what's even more interesting, I would say, is that there are ones, oh, this is cut off. There's a peak there, I promise. There are ones that are dibrominated. And so that, to me, was really fascinating because there was this bevy of uncharacterized natural products from this organism. To date, there were no reported dibrominated alkaloids from Cleona. So I think already we're kind of starting to get into something that's relatively interesting. The mass doesn't appear to be a simple second bromination of an already known compound. So it might be a relatively new scaffold as well. And finally, I wanted to see if, sometimes you get a sponge, especially because a lot of the sponge we keep in an aquaria with a lot of other organisms. Who knows what kind of crosstalk we might have? Maybe somebody gives a bacteria to somebody else. Maybe it's not a natural phenomenon. It's just kind of a one-off. How widespread is this? And so Samantha and Vivian endeavored to collect various samples from various sites across different times of year. And in doing so, oh man, this is really messed up. Okay, that's okay. There are peaks there. And you can see some of them now. So this peak G is the dibrominated metabolite, but these metabolites are relatively conserved across multiple samples, across multiple times of year. And so this isn't a seasonal effect. This isn't an individual sponge. This is something that we think is kind of native to this organism's biology. So I thought this was really fascinating. And so we went to try to isolate these molecules like any good natural product chemist would. So we got a big sponge, gave it our fine dice treatment, and put it into methanol. And we saw that we were able to, again, in LCMS, identify all of those compounds. And I tried to purify them. And every time I tried, they degraded. I tried normal phase, reverse phase, LH20, countercurrent chromatography, which is something that probably most people here have never heard of. And I had to dig it out of the basement in Scholander Hall. And so I tried everything I could try to get my hands on. And this consistently resulted in decomp. And again, part of why I should have read the literature a little more deeply rather than looking at the pictures and seeing if the molecules matched was because somebody already knew this. Somebody already figured out that no matter how hard you try, you're going to get nothing if you just try and purify it regular. And so in order to characterize these compounds, as well as the related compounds, most of the time you need to derivatize them. And so if you derivatize the extract, you get maybe a little bit more stable set of molecules. In my hands, they're really not that much more stable, but at least you can isolate a little bit of them. And through doing so, we were able to isolate this really interesting molecule, which we call Andersenamide A, named after Raymond Anderson, who isolated some of the first sets of these molecules from British Columbia. And so this was a lot of fun. We got to spend a lot of time. We found one molecule through a lot of hard work and derivatization. And I saw so many more. I saw so many more compounds in this extract. And I didn't want to do this again. And it wasn't easy. Isolating these molecules wasn't straightforward. They're produced in extraordinarily diminishing small d. What can I do to try to understand what the true biosynthetic potential of these organisms is? And so I did my best and tried to generate a molecular network, and I was immediately overwhelmed. And it was extraordinarily challenging for me to be able to look at a nested molecule that I knew and look at its surrounding nodes and say, oh man, like, do I even know if this other compound is brominated? And I could go back and forth and try to figure this out. And I presented this wonderfully. Peter invited me to a group meeting at his lab, and there were two wonderful postdocs there at the time who told me, you're doing this the most difficult way you can possibly do this. And that we developed this brand new tool called MassQL, where you can curate your data set, where you build a query to say, can you find me ions that have this specific feature? And then can you select those ions and generate new molecular networks so I can maybe interrogate them a little bit more targeted? Okay, great. Okay, so doing so, we built a query for dibrominated metabolites.
I think you sent me a truncated presentation.
Oh no. Okay.
We have a solution. We have a solution.
You want me to hit my computer?
No.
Oh no.
Yeah. I believe you do chalk talk.
If you ever decide to apply to a PUI, what they don't tell you is that you have to give 2 talks. Well, I'm telling you now, I guess. You have to give 2 talks. You give a research talk where you do kind of some of this, and then you have to give a chalk talk where usually you have to teach a lecture or maybe even do it on the board. And so with great unfortunateness, I will now do this for you today.
Thank you. Not scripted. This is not.
I will, in fact, never forgive you for this. And so what we saw was that using this, we found a network that formed along with this molecule, which was androstenamide A. And it had a bunch of things connected to it. So this is the base structure of our compound. I now had a series of real nodes, if this was this node, for example, that were connected to this compound that I could investigate in like a real way and know that they're actually related to this molecule because they're already pre-selected to be dibrominated. One of these nodes, get in trouble. One of these nodes was a molecule that looked like this. Okay. So one of the nodes was a carboxylated version of this molecule. And what I didn't tell you about the biosynthesis of some known enamides is the reaction to generate a decarboxylated version is you lose CO2, which would be this part, and then you need to lose these 2 hydrogens, then that gets you the double bond here. And so through using MassQL, you're actually able to identify a putative biosynthetic precursor that also was suggestive of the order of reactions that happen in the construction of this molecule. First, you build the peptide, then you hydroxylate it and brominate it. Then the final product is the intermediate is decarboxylated to give you this final product like this enamide. And so not only was MassQL super helpful for me to just be able to curate my data, but it actually allowed us to generate a useful and interesting hypothesis that we'd be able to test to understand the biosynthesis of these molecules more. And so I'm going to go to not my summary slide because it doesn't exist, but to the slide with my undergrads on it because they're really the ones who did the vast majority of this work, along with some wonderful collaborators who I have and hope to keep collaborating with. And, there's a lot of really cool future directions for this. Like I said, there's a ton more molecules here. We're really excited about trying to figure it out. There are a lot of these enamides out there in the literature that are broadly distributed in the marine environment. No idea how those are made. Are they made the same way? Are they made differently? Can we apply a similar methodology to identify congeners? And finally, it'd be wonderful to try and use something like native mass spec to see if these things are actually chelating molecules in nature and what the ecological significance of those could be. And I think with that, I think I'm just about out of time, and I'm more than happy to take any questions. No forgiveness.
Yeah. Well, first of all, thank you, Vic, for being such a good sport. The real trick is, it's unpublished, so nobody on Zoom saw it. Anyway, any questions from the audience?
Please. Very nice talk. And someone who's worked with sponges, I can agree with you. They are gross, but beautifully gross. So my question, I had two questions. So first was, I think you mentioned in the beginning of your slides that these are like vanadium chelators. Is that something you think is needed for the halogenase? Because a lot of these halogenases are vanadium dependent. So that's something relation over there?
Yeah, that's a great question. And I wish I could say yes. But the truth is the vanadium, the one that we think is a vanadiphore, the molecules aren't halogenated. And so I don't know if that's part of the role. Another thing that people think these molecules do is in insects, you have these polyhydroxylated aromatics that form the skeleton in the sclerotization process. And it's an oxidative process. It uses a lot of interesting oxidative enzymes to cross-link phenolic groups. These molecules are found in the cape of the tunicate, and people think that that might be a part of wound healing, is that when they get cut, they're full of vanadium, which can catalyze oxidative chemistry, and these things might stitch together through cross-linking that's oxidative. Again, not a lot of people have really had the chance to investigate the role of these molecules, but this is the guess that's out there.
Yeah, so that's a good. The second question was, you mentioned that you think these could be made by the host itself and not be microbial because of the diversity you see. Are you planning to do like any metagenomics study on this? Or?
Yeah, one of the slides that got cut off was I did a very shallow shotgun metagenome assembly, and I didn't have long enough contigs to really get anything conclusive. But what I was able to see is that there's a large diversity of halogenases encoded within the metagenome of this organism. And if you build the phylogenetic tree, you'll see that many of them cluster with the very canonical tryptophan halogenase, as well as some of the RIP halogenases. A RIP is a ribosomally synthesized, post-translationally modified peptide synthesized by the ribosome, and it usually gets made, and then it gets halogenated post. So there are also halogenases in the metagenome that also play with those RIP halogenases as well. So maybe, but they're all bacterial looking. So it's not clear to me if there is an animal halogenase in there. And as far as I know, there are not very many known animal halogenases outside of ones that are non-canonical, like the ones that are used to make your thyroid hormones. Yeah.
There's a question.
Please.
Hi, my curiosity is mostly you find the compound that you brominated compound, but I'm just curious, do you also find S-related compound, but it's also chlorinated?
Yeah. So you're asking if I find like the non-halogenated ones or the chlorinated or iodinated ones? Yeah. I don't see this. I don't see differentially halogenated variants. A lot of people suppose, right, there's a lot of cases where a halogenase can only brominate due to redox potential of the enzyme. And so I think that these might be the case that they maybe only are capable of brominating to oxidize bromine instead of chlorine. Unclear though. Good question. Yeah, please.
A question from the Zoom. We also have 30 or 40 people watching on Zoom. What's the specific property of bromine? So it's kind of like incorporated here selectively. Do you have any idea what the effect of specific bromine is?
What the role of bromine is a very difficult question for me to answer. Yeah, no idea. A lot of people think it's for solubility. It's very often found at the C6 position on the indole. As you can see, kind of not in these ones, but if you look at other natural products, you'll find that this is the most commonly functionalized position, especially from sponge natural products. But no, I don't think I can assign or ascribe a discrete role for why they're brominated other than you find a lot more halogenated metabolites in the ocean compared to on terrestrial ground. A lot of the time we just hypothesize because there's more salt in the ocean. So that might be the simplest reasons I can give.
Thank you.
Yeah, please.
Fascinating talk. So have you thought about a lab system where you could potentially express these enzymes and have like a more tractable sort of a platform to work with?
Yeah, what a great question. These for heterologous expression, probably not benthy, I don't think. But a lot of the times people will be able to, a lot of the proteins, especially the ribosomally synthesized peptides, are frequently intronless. And so you can use a bacterial host that works quite nicely. For the other enzymes in the Moore lab, I worked with yeast quite a bit, and particularly expressing animal proteins, and it works quite nicely. So depending on your choice of host in yeast, whether you use Pichia pastoris or Saccharomyces, you usually can find a host that can express an enzyme that you're interested in.
Okay, let's give Vic another round of applause. You really showed your teaching chops. We have coffee right now, and we'll have the keynote in 20, 25 minutes. So mingle.